Hello and welcome to our short talk summarizing our products and services. I'm Anna Lisner and will give you the talk today. So let's start. So here we have an overview of all our products and services. All our products are highly complex and optimally designed oligonucleotide pools. And this basically means is that one product consists of several oligos that are targeting the same gene or transcript. For example, the siPOOLs, our gene silencing tool, consists of 30 siRNAs that are all targeting the same gene. And the riboPOOLs and the raPOOLs are a pool of biotinylated oligos. And the riboPOOLs are used for ribosomal RNA depletion of any species and depletion of any abundant RNA. The raPOOLs are used for enriching RNAs and their interacting proteins and nucleic acids to understand and study what the RNAs are interacting with. And we also offer services, and most services are all around our core expertise of RNAi, such as big data analysis suite for RNA and CRISPR screens. And we also offer RNA validation projects, RNAi screens, and transfection efficiency services. We also offer NGS services all around our riboPOOLs. And yeah, before I will go into detail about our products and services, I wanted to explain to you the pooling approach we use for all our products, as I've said before on the next slide. The power of pooling is best explained with single siRNAs versus a complex pool of siRNAs. But the concept of pooling can be applied to any kind of targeting, such as targeting ribosomal RNAs with our riboPOOLs. So here on the left-hand side, you can see a single siRNA that was designed to knock down a gene, shown here in green, and to achieve a good knockdown of that gene, high concentrations are necessary. And what high concentrations lead to is non-specific targeting of genes that are not supposed to be targeted, and not supposed to be knocked down. And this phenomenon of knocking down other genes, other than the gene that is supposed to be targeted, is called the off-target effect. And actually, the company siTOOLs Biotech has been founded to conquer the off-target effect. And here on the right hand side, you can see how we siTOOLs Biotech conquer the off-target effect. We pool 30 siRNAs in one pool called the siPOOL, and this enables more specific targeting, as each of these 30 siRNAs has a different sequence and is targeting a different part of the gene. Moreover, the concentration of each of these siRNAs is reduced to a minimum, and this is how we kind of dilute away the off-target effect, and therefore the power of pooling enables higher specificity and higher efficiency and better reproducibility of experiments. In other words, you save money and time using pooled products. And as I said before, the pooling approach is also used on our other products, the riboPOOLs and the raPOOLs. So let's check out some data of how the siPOOLs actually do lower the off-target effect to a minimum. Here on the left-hand side, you can see how a single siRNA changes the expression pattern of numerous genes. So the siRNA, the single siRNA, was designed to knock down a gene called SKIL1. And the knockdown of that gene is efficient, which is shown here in green. But the knockdown also led to a change of expression patterns of other genes, which is shown in dark red. So each of these dark red dots represent a off-target. And when the same siRNA that was used on the left-hand side is part of a 30 siRNA pool, and all of these 30 siRNAs are designed to knock down SKIL1, the off-target effect is much, much lower. Whilst the knockdown of SKIL1, of the gene that is supposed to be knocked down, is efficient. And this means that the siPOOLs are a great tool for gene silencing, because it is highly efficient and robust and reliable. The take-home message is, if you want to do any RNAi experiments, use siPOOLs to save money and time. And the next tool I'm shortly presenting to you are the riboPOOLs. RiboPOOLs are a molecular tool for ribosomal RNA depletion and abundant RNA depletion. As I mentioned before, all our products are pooled oligos, and the RiboPOOLs are a pool of biotinylated single-stranded DNA oligos, and the RiboPOOLs are antisense to ribosomal RNA. As you probably know, ribosomal RNAs make up to 80 to 90% of the transcriptome, so ribosomal RNAs are very abundant and would lead to very high cost of RNA-seq experiments if not removed prior to RNA-seq. And yeah, this is how you can remove ribosomal RNAs with riboPOOLs. You include the riboPOOLs to the mixture of relevant RNAs, the RNAs that you want to sequence, and the ribosomal RNAs that you want to deplete. And the riboPOOLs hybridize to the ribosomal RNAs. And its riboPOOLs are biotinylated. You can use streptavidin-coated magnetic beads to capture and remove the ribosomal RNAs. So easy peasy RNA removal, which takes approximately 70 minutes and is enzyme-free. And we offer the riboPOOLs for all standard model organisms and many more, as we can design a riboPOOL for any species. We also offer pan-riboPOOLs, which can be used on many genera from one phylogenetic class or a whole phylum. And yeah, check out more about our riboPOOLs on our riboPOOL site. As I said before, the riboPOOLs and the raPOOLs are chemically similar. The raPOOLs are also a pool of single-stranded DNA oligos that are biotinylated. The pooling approach enables higher target coverage and therefore higher specificity and higher efficiency. The raPOOLs are used to enrich RNA and their interactome. This enables studying the properties of targeted RNAs and understanding what the RNA is interacting with. The proteins and nucleic acids that are interacting with the targeted RNA are cross-linked before cell lysis. After cell lysis, the raPOOLs are included to the lysate, and the raPOOL hybridizes to the targeted RNA. The capture of RNA and its interactome is done using streptavidin-coated magnetic beads. And after elution of proteins and nucleic acids, you can analyze interacting proteins and the nucleic acids. And you can learn more about our raPOOLs on our website. And last but not least, our services. Our bioinformatical service is basically a service that makes sense of RNAi screening data. We have a data set or a database of many siRNA screens in numerous cells, and we can help you find the on and off targets of siRNAs and therefore help you identify, for example, new cancer targets or pathways. We also offer research services all around RNAi. We can help you set up a RNAi screen. We can help you with optimizing the transfection protocol that you're using for your siPOOLs or siRNAs. Or we can validate any siPOOLs for you. Moreover, we offer services around our NGS tools, the riboPOOLs, and we can help you optimize the rRNA depletion efficiency, and we can also perform the rRNA depletion for you, and this is subsequent library prep and the sequencing. Thank you very much for your attention. In case you have any questions, just write us a mail at info@sitools.de or ask your local distributor. And if you're not convinced of our products and services, big pharma like Roche, J&J, and Bayer are already our customers. So why shouldn't you?